back to the crash course in bioinformatics. This week we're going to be talking about BLAST, not this one, but this one, the Basic Local Alignment Search Tool. The BLAST, or otherwise known as the Basic Local Alignment Search Tool, it is a main NCBI tool for comparing a protein or DNA sequence to other sequences in various databases. This tool finds regions of local similarity between proteins or nucleotide sequences. The program then compares the protein or nucleotide sequences to sequences in a database and then calculates the statistical significance of the matches. A typical BLAST reveals what related sequences are present in the same organism and in different organisms as well. A BLAST search allows its users to select one of the sequences, which is known as a query sequence, and then performs a pairwise sequence alignment between the query and entire database, that is the target database. And then tens of millions of sequences are evaluated in a BLAST search. Just exemplifying a typical DNA database, 50 billion nucleotides are searched against, and only the most closely related sequences are then returned. So let's just see what are the basic steps in which a BLAST search is performed. So the first step involves selecting a target sequence. Then after the target sequence is selected, we basically decide upon the exact BLAST program that we need to use. The next step is to decide upon a desired database, and then finally we can select our optional parameters for the search and as well as for the output. And this is how a search or a BLAST search is completed. So let's look at all of these individually. So the first step, obviously, it is specifying the sequence of interest, and this can be done in one of two ways. So over here I've just given an example of a basic local alignment search tool. I went to the page and then we started off with a BLAST-P. And over here in the query sequence we can enter either FASTA sequences of the proteins or genes that we want to enter. For example, over here I'm entering the protein sequence of hemoglobin subunit beta. I searched for its protein sequence, and then when the page that gave me its FASTA format for the protein sequence, I simply took that sequence. From that page I copied it and I pasted it up as a query sequence for my BLAST search, like so. Alternatively, what we can do is we can use excision numbers for our BLAST search. It's a bit more convenient on part of the user to use the extension numbers if they are known. So I can simply use the extension number that I took from a paper, or I can simply copy it from the search, and this is going to give me the same results as pasting a FASTA format sequence. So just specifying the sequence of interest, we can either have a FASTA format sequence or we can have a sequence that is retrieved from NCBI. The next step over here is to decide upon a particular BLAST program, and there are 5 different types of BLAST programs that are offered: BLAST-N, BLAST-P, BLAST-X, T-BLAST-N, and T-BLAST-X. The BLAST-P and BLAST-N are the most commonly used ones, and the BLAST-P being a program that compares a protein query to a database of proteins. And similarly, the BLAST-N is a program that compares a DNA query to a DNA database. So these are a little bit simple. But the rest of the 3 programs, they are known as translated BLAST. So what are translated BLAST, and what is the principle upon which they operate? The principle is basically the concept of 6-frame translations. So basically, the translated BLAST, they use the fact that DNA sequences can be transcribed and translated into 6 potential protein we can say potential reading frames. Because any DNA molecule encodes protein sequences by a series of 3 nucleotides, right, which are known as codons. So any given sequence of a DNA can therefore be read in 6 different ways. So 3 reading frames in one direction, starting at a different nucleotide, and then 3 in the opposite direction, as we can see in this diagram over here. The BLAST program hence searches the user genetic code to translate either the query database subjects or both into protein sequences, which are then aligned in a BLAST-P manner. The translations are performed in 3 forward as well as 3 reverse reading frames, so that no possible translation it is mixed. So 6-frame translation, it is used in all of these 3 programs, the BLAST-X, the T-BLAST-N, and the T-BLAST-X, but in different ways. In the tblast X, basically the comparison of a nucleotide query sequence, it is done by translating that nucleotide query sequence in all of the reading frames against a protein sequence database. So for example, if you have a DNA sequence and you want to know what protein it encodes, you can perform a BLAST X search, and this automatically translates the DNA into 6 potential proteins, and the BLAST X program then will further continue to compare each of these 6 translated protein sequences to the members of a protein database, and then give you your result. The tblastn, however, compares a protein query sequence against a nucleotide sequence database, which is then also translated into all of the reading frames, and one can use this program to ask whether a DNA database encodes a protein that matches your protein of interest or not. If you have a protein and you want to know regarding its sequences, so you can perform the tblastn. The tblast X compares the 6' translations of a nucleotide query sequence against 6' translation of a nucleotide sequence database, as it is also seen in the figure, and this is the most computationally intensive BLAST. For example, consider a situation in which you have a DNA sequence that has no obvious database matches, and you want to know if it encodes a protein with a distant, statistically significant, however, database matches in a database of express sequence tags, for example. So a BLAST X search would be more sensitive than a BLAST N, and therefore useful to reveal the encoded proteins that are homologous to your sequence series. So this is the 5 types of BLAST programs that are offered, and we can search our desired sequence to either a DNA or an RNA based upon all of these different programs that are offered by BLAST. The next step is to basically look for a particular database, and there are different databases for proteins as well as nucleotides. So the protein databases, basically the default option is a non-redundant database, and what non-redundant databases are, these are derived by merging many protein and DNA databases, and these databases often contain identical sequences. Generally, only one of the sequences is retained by the non-redundant database along with multiple accession numbers. So the non-redundant databases are often considered to be one of the best sites for searching of the available sequences. Other options for the proteins are the reference protein databases. So for the DNA database searches, we have a lot of other options available for which consist of sequences from a lot of other sources, for example, GeneBank and EMBL and the Databank of Japan. And otherwise commonly used options include the human or mouse genome plus transcript databases or ESC databases, and these are other databases that are most commonly used for the nucleotide sequences. So selecting the database is basically up to you, whatever you want to do. The best database, you can decide upon what you want to find out from a BLAST search. The next step, it is to select the optional parameters, and these parameters may or may not be tweaked with regards to what you want to search for. So the first thing that we have over here, it is maximum target sequences. So the maximum number of target sequences can be set to 100 by default, or you can increase or decrease it according to your will, but the default parameter of the maximum number of target sequences is considered to be 100. The next in this is short queries. So if we select this option, the option of the expected value and the word size are automatically adjusted, and you don't have to worry about adjusting them later, but they can also be adjusted afterwards. So short queries is just a quick way of adjusting the parameters automatically. The next, it is the expected threshold. So the expected value or the e-value, and we're going to be talking about e-value loads in the next section. So the expected value is basically just the number of different alignments with scores equal to or greater than some scores that are expected to occur in a database search by chance. So basically a probability. So a reasonable general guideline, very basically over here, it is that any database search or having e-values of 0.05 has statistically significant. So we're going to be talking about e-values more, how it is calculated and what are the algorithms of that. So, but the basic general guideline, it is illustrated in front for now. The next thing is basically the word size. So what basically the word size is, that is whenever a query is used to search a database, The BLAST algorithm first divides the query into a series of very small sequences which are known as words of a particular length, which is called its word size. So for the BLAST tree, a larger word size yields a more accurate search. So for any word size, matches made to each word are then extended to produce the BLAST output. So this is basically how BLAST works. So in practice, the word size can remain at 3, that is set at a standard, and should be reduced to 2 in case of a BLAST tree only when the query is very short peptide sequences. But generally the word size, it is by default set at 3. For nucleotide searches, the default word size, it is 11, and it can be raised to like 15 or reduced to like 7. And lowering the word size for a nucleotide BLAST yields more accurate results, but they're going to be a bit slower, but they're going to be more accurate. The next is the maximum matches in a query range. So matches to one of the regions of interest can be obscured by frequent matches to a different region of the protein. So this feature basically will offer a solution in which the redundant databases hits are just discarded, and to get a very clearer result of what we are trying to find. The next is matrix, and we've discussed about the matrices, the backhand of the operation earlier as well, but by default over here 8 amino acid substitution matrices are available. Other than that, different other programs, alternative programs, they offer a much more choice of substitution matrices. So it is sometimes advisable to try a BLAST search using several different scoring matrices to just see what goes around. The next thing is to look at the gap costs, and we've talked about gaps. So just as a review, a gap is basically a space that is introduced into an alignment to compare for insertions or deletions in one sequence relative to another. So since a single mutational event may cause an insertion or deletion of more than one residue, the presence of a gap is frequently ascribed more significantly than the length of the gap. So the gap introduction is therefore penalized very heavily, whereas a lesser penalty is ascribed to each subsequent residue in the gap. To prevent the accumulation of too many gaps in an alignment, introduction of a gap causes the deduction of a fixed amount from the alignment score. The extension of the gap to encompass additional nucleotides and amino acids is also penalized in the scoring of an alignment. So we talked about the gaps. We can basically adjust the gap costs according to our will, but there are default settings and we can also use them. But the concept of gap is very much clarified in the previous discussion as well. Then we have compositional adjustments. So the basic or the default setting, it is the conditional computational scoring matrix adjustment, and it basically just at the backhand, it helps in the calculation of e-value statistic. However, if the proteins with non-standard compositions are seen, the computation adjustments are made in order to increase the accuracy of the BLAST search, but this is done when we really have proteins that are non-standard in composition, and then we have to tweak these values. Otherwise, the conditional compositional scoring matrix adjustment, it works just fine. And next, we basically have the filter. So what filtering does, it basically masks the portions of a query sequence that have low complexity, which can be sequences in the form of strategies of amino acids with limited information in their content. For example, dinucleotide repeats of like CA repeating nucleotides, or we can also go for ALU sequences or regions of proteins that are extremely rich in one or more than one amino acid. Similarly, some stretches of hydrophobic amino acid residues that form a transmembrane domain are very common, and a database search with such sequences results in many database matches that are statistically significant, but while they have no biological relevance. So other motifs that are masked by filtering include acid, basic, and protein-rich sequences. So we can basically use these filters in order to clear out the clutter and just to focus on the target sequences that basically make sense. Last but not the least, we have masking. So the mask for lookup table only, it basically it masks the matching of words above threshold to database hits. So this basically avoids the matches to low complexity sequences or repeats, and BLAST extension then occurs without masking. Okay, and then the mask lowercase letters option allows you to enter a query in a faster format using uppercase characters for the searches, but filtering those residues you choose to filter by entering them in lowercase. So these options have little effects on majority of the blast searches that we perform, but still they can be tweaked in order to get a better blast search. So just as a practice, we can over here observe the blast search that I performed for the SOX2 protein in human beings. I searched for the protein and then the blast query popped up. So they basically put in the extension number for the blast query. Assigning a job title, it's always a good practice, and then over here we can choose different databases we saw over there, because it's a protein and we are going for non-dependent databases. The organism that we want to search against is the rat, so I just chose the rat tested, and these are all of the other additional parameters that we just discussed, and we can change them. For example, the maximum number of target sequences we can alter, the word size we can change. It is already adjusted to a 28, so we didn't mess with that because we didn't need to, and then we basically perform the blast search. So over here what it is going to basically give us the results of the SOX2 gene in the human beings to its counterparts if they are present in rats. So this is basically the result window that we obtain, and the results appear in this way. So we can see over here a lot of information, most of which we know by now. Over here, if we go to alignments to get just a better view of what the results have to say for us. So at this particular hit, we can see the number of identities. So what identity is that if there were there are about 2,243 out of this required number of sequences that are exactly the same. So I can say that this sequence of human SOX2 protein is 89% identical to the sequence that we searched for in the rat. And then we also have a concept of gaps of the areas where this sequence differs, and we have various plus signs also available, not seen right now, but the plus sign they indicate a substitution event in which the sequences in which the change of the amino acid does not have a very big impact on the functional effects of the protein. So this is just a general overview of how a blast search can be done to the point of getting the results. So the next discussion about interpreting the results and finding out the best blast hit, it is going to be carried out in another video. Thank you so much for watching, and I'll see you guys in the next video. Bye.